DNA fingerprinting is a forensic technique used to identify individuals by characteristics of their DNA. The technique relies heavily on the principle that no two individuals have the same genetic code. As we all know, 99% of the genes in any individual remains same, but the difference lies in a short sequence of repetitive DNA that is different in each and every individual. So, DNA fingerprinting relies on the sequence polymorphisms. What do we mean by sequence polymorphisms? They are usually single base pair changes that lie in the repetitive DNA among different individuals. Now, we see how is the technique carried out in a forensic lab. The steps include collection of samples, isolation of DNA, fragmentation of DNA, separation, Southern blotting technique, hybridization, autoradiography. Finally, the last step is the comparison of the different samples. To begin with, the first step includes the collection of the samples. For DNA fingerprinting, blood, hair, saliva or even nails can be used, as the DNA remains same in all the cells of an individual. After the collection of the samples, we begin with the isolation of DNA. So, here we are using a blood sample for the isolation of DNA. The first thing that is done is to take the sample in a small tube called the microcentrifuge tube or the MCT. After taking the sample in the MCT, a detergent called SDS, that is sodium dodecyl sulfate, is added to the sample. What is the purpose of adding the sodium dodecyl sulfate? As we all know, DNA is contained within the cells. So, for releasing the DNA out of the cell, this detergent is added. This detergent, it is an anionic surfactant, so it breaks the interactions among the membrane proteins and lipids. As a result, cell lysis happens. So, for allowing the cell to lyse, we add some microliters of the detergent to the sample and incubate the sample at room temperature for some time. So, in this time, the cell will lyse and the contents will be released. After that, we centrifuge the sample along with a balance in a centrifuge. This gives us two fragments. The white part that you see is the precipitate, and then the clear solution on top is the supernatant. The precipitate basically contains the cell debris and the DNA comes out in the solution. Our next step is the precipitation of DNA from the sample. So, how do we precipitate the DNA? At room temperature, isopropanol works best for precipitation of nucleic acids, because nucleic acids are insoluble in isopropanol. So, again we add few microliters of isopropanol to the sample and we incubate the sample at room temperature for some time. So, after adding isopropanol, the DNA precipitates out in the solution. Now, we will incubate the sample for some time. After incubation for some time, we centrifuge the sample again. After centrifugation, we remove the solution, and what we have got as the white part is the precipitate containing our DNA. The next step is giving the DNA an ethanol wash. This wash with ethanol is very important to remove all the other impurities and salts from the DNA to obtain our DNA in pure form. After giving the ethanol wash, it is again left for some time, and here you can see that the DNA has precipitated. Now, this DNA that we have got, it is the genomic DNA. It is huge in size. So, for the purpose of DNA fingerprinting, we need small fragments of DNA. For that purpose, now we have got our DNA, we add an enzyme which is called the restriction enzyme. So, what does the restriction enzyme do? We will add the restriction enzyme and incubate the sample for some time. After this, we will find that a huge chunk of DNA has been broken down into many smaller fragments. That is the function of the restriction enzyme. It is also called restriction endonuclease. They cut the DNA at specific sites within the DNA, not on the outside. Within the DNA sequence, the restriction endonuclease cuts the DNA, and the recognition sequence for each enzyme is different. So, after treating with restriction endonuclease, we get a pool of DNA of many different sizes. Our next step is that from this pool, we have to separate the fragments. It is just a mixture. So, we want to separate it out. For that purpose, we do a gel electrophoresis. The gel electrophoresis technique helps us to separate the DNA fragments according to their size. So, for that purpose, what we use is an agarose gel. In the agarose gel, the DNA moves and it separates according to the size. But how does it move? You can see here on the top of the gel there are wells. These are used for loading the samples. These are called wells and are used for loading these samples. You load the DNA extract each sample in each well. After that, you fill the tank with a buffer and connect it to a power source. Now, if you see here, there is a cathode and an anode, and we know that DNA is negatively charged. So, when you load your samples in the well here, DNA will move from the negative pole towards the positive pole, and they will migrate according to their size. What do I mean by that? The larger fragments will move very slowly in the gel, while the smaller ones will run through the gel and separate out. As you can see here, the smaller fragments will move further down the gel and the larger fragments will remain towards the upper end. So, the DNA fragments from a pool can be separated according to their size in a gel electrophoresis. The DNA fragments are separated according to size. The next step, after separating the DNA fragments, we want to identify a particular DNA fragment. How do we do that? That is usually done with the help of the Southern blotting technique. This technique has been named after its discoverer, E.M. Southern. So, we will now see the assembly for the Southern blotting technique. This is your gel containing the different separated fragments of DNA. For the Southern hybridization, first we take a tank and fill it with an alkaline solution. On top of that, we put a sponge. Above the sponge, now you will place your gel. Now, from this time onwards, whatever fragments of DNA that are here, you call them as target DNA. You call them as target DNA. On top of the target DNA gel, you place a nitrocellulose membrane. Whatever DNA fragments are present in the gel, they will be transferred onto the membrane. On top of the membrane, you put a stack of blotting sheets, and the size of the sheets should be equal to the size of the gel. On top of these blotting sheets, you put a weight, and that weight also should cover the blotting sheets evenly. Now, if you see the assembly properly, you will be able to make out how will the transfer occur. The transfer will occur due to the capillary action. The buffer will move from the tray through the sponge, through the gel and on to the paper towels. Why does that happen? That happens because the water is moving from higher water potential to lower water potential, that is your blotting sheets. And by this capillary action, the DNA from your gel will be transferred onto the nitrocellulose membrane. One more very important thing is here, that the solution or the buffer that we use for transfer, that is an alkaline solution. This alkaline solution will denature our double-stranded DNA into single-stranded fragments, and only single-stranded fragments are transferred onto the nitrocellulose membrane. This is the direction of the capillary action. Onto the nitrocellulose membrane, because of this alkaline solution, this double-stranded DNA will be fragmented into single-stranded DNA and it will be transferred. After the transfer, on the membrane, the exact similar pattern of DNA as that on your gel will be produced. Now, how will the identification of the particular DNA fragment take place? That will happen because of the next step, that is hybridization. Hybridization is the process of forming double-stranded DNA molecule between single-stranded probe and single-stranded target DNA. So, for the hybridization to happen, your membrane contains the single-stranded target DNA. You will dip the membrane in a solution containing radioactive probe. What is the probe? A probe is a piece of single-stranded DNA of identical sequence to your target DNA, and it is labeled with a radioactive substance for the purpose of identification. So, you add the probe to the buffer and you dip your nitrocellulose membrane in that buffer and leave it for some time. So, the probe forms a complementary base pairing with your target DNA wherever it identifies a similar sequence. This is how in the alkaline solution your double-stranded DNA will open and form single-stranded DNA like this. Now, you add the single-stranded probe. The probe forms a complementary base pairing with the DNA and again a double-stranded DNA is formed. Out of this, one is your target DNA and the other is the probe. Now, the annealing of the probe has happened. So, after that you remove the excess probe that is there so that you get a clean result. For the detection of the probe, the membrane is placed in intimate contact with an X-ray film. It is placed in intimate contact with an X-ray film, and after developing the film, wherever the probe has bound to the target DNA, you will develop black patches. These black patches show the presence of the target DNA, identical to the probe. Now, you can repeat the same experiment for as many samples as you wish. The same experiment at a crime scene can be repeated for different suspected individuals, followed by comparison of the sample from the scene of crime and the suspected individuals, and based on the results very easily you can identify the criminal.